Okay, so I guess I didn't finish the biotechnology lecture yesterday, so I figured I'd try to wrap up a couple of loose ends. One of those is gel electrophoresis. I guess we talked about PCR, but we didn't quite get to gel electrophoresis, so I figured I'd talk about that today in context of PCR and what it can do and so on and so forth. Yeah, so I guess I usually start these things out with a little music. Yeah, so let's go with here.
There was a time when to amplify DNA, you had to grow tons and tons of
What is
tiny
this?
cells.
Is that Kenny Rogers? Do you mind? I'm trying to listen to the song here. Oh, sorry.
Said you can amplify in vitro.
Oh, that's Bob Dylan.
Just as well.
He's got a Nobel Prize, yeah?
Just fix your tablet with
Oh, look, there's Mariah Carey.
some primer.
Dude, didn't I just tell you to be quiet? Oh, look, there's Hall & Oates. Dude, be quiet. I told you to be quiet. Hey, Stevie Wonder. Look, I'm not going to tell you again. You need to be quiet. Look at the boss.
Well, it's
Bruce
amazing what
Springsteen.
heating and cooling and
Cut it out.
heating will
Oh,
do.
man. Wow, it's just like that Live Aid thing. Oh, look, it's Geddy Lee. Oh, boring. Anyway, so sorry about that. So PCR and gel electrophoresis. I hope you enjoyed your musical introduction. Sorry about the tussle there. That guy just wouldn't shut up. Anyway, so PCR works like a DNA Xerox machine to make billions of copies of a single segment of a genome, and you can use that DNA, that little amplified section, for diagnosis when you need to detect mutations, when you need to solve a crime, or find out about the paternity of a potential person. Forensic investigation. You can also use amplified DNA in the same way that we discussed in class for taking that piece of DNA and putting it somewhere. You could amplify the GFP sequence or the insulin sequence or any sequence like that, and then put it, after digesting with restriction enzymes, putting that segment into a plasmid and then transforming a bacterium with it. So those are things you can do with PCR. It's an extremely widespread and useful technique. And again, we talked about how we can heat DNA to break the hydrogen bonds between the double strands. We use primers. Primers match the sequence perfectly, right? If the beginning of the sequence is ACTG, then we can make a primer of ACTG that binds to the opposite side and is copied in this direction with DNA polymerase, Taq polymerase from Thermus aquaticus bacteria. That is a heat-tolerant bacteria that you can find in Yellowstone parks, hot springs, and so on. Prismatic spring. We get another primer that matches the very end of our sequence, and the same enzyme can copy in that direction, and in that way we only copy the target sequence. We start here, we end here, and we have zillions and zillions of molecules of DNA. And as long as our primers are 15 to 30 base pairs long, then we could never bind to the wrong place by bad luck, because the sequences can be, again, with 4 to the 30th power is effectively a unique sequence, that the chances of another sequence matching that perfectly in that genome is less than 1 in a million. So again, we heat, we break the hydrogen bonds, at temperatures of around 94 degrees Celsius, very nearly boiling. This is not hot tub temperature. This is cook your lobster temperature. You cool to about 55 degrees Celsius. This is still slow, painful death temperature, not hot tub temperature. And the primers bind, and then we heat to the ideal temperature for Taq polymerase, the DNA polymerase, and then Taq polymerase grabs the primer and makes a copy of DNA using little nucleotides, little pieces of A, C, T, and G, copying from the primers, yeah? And once it's done, we've taken one strand of DNA, we've made two, and then we can go back to the very beginning. There's another video that explains this in more detail than I'm providing, and I think it's this one here. It's a good video to watch. I won't show it, but I recommend that you watch it. You'll learn and understand better. Sadly, some people do a better job than me. All right, so heat the DNA, get RNA polymerase to copy from the site of the bound primers, and after 3 replications, you heat and cool 3 times. You start with 1 piece of DNA. You should end up with 2 after the first round, 4 after the second round, 8 after the third round. So your answer should be C here. Okay, and again, we can use DNA to diagnose or to look at that. Once we've amplified genetic sequences, we can look at them. We can look at living people's DNA. We can look at dead people's DNA, and we can see if those people are related. And we can use that to follow the migration of modern humans, starting in East Africa and migrating all over the planet. So, gel electrophoresis. How do we use DNA in diagnosis? Well, gel electrophoresis can do it. The DNA of 2 people of the same gender is 99.9% identical, right? That's 1 difference in 1,000. You have 6 billion base pairs. 1 difference in 1,000 is still 6 million differences. So humans are very, very similar, and yet each person is still unique. And we can use restriction digestion and gel electrophoresis in combination in order to diagnose disease, genetic disease, in order to diagnose infectious disease by looking at bacterial DNA instead of human DNA, or to solve paternity disputes or whatever. So once we amplify DNA using PCR, we can analyze DNA, analyze the amplified DNA using restriction enzymes and this technique called gel electrophoresis. So DNA sequences of different length are easily measured using gel electrophoresis. So here's the technique. We use a gel, kind of like Jell-O is a gel, but this is agarose gel. Agarose gel is, here's a picture of agarose gel. I'm sorry, I should have these in better order, and I don't. We purify that from seaweed. If you've ever eaten high chews, they have agar in them. Agarose is very purified agar. And it's a gel. It's porous at the molecular level, and the DNA moves through the pores. DNA, and so when we take this gel, we would expect DNA to be able to diffuse through it, albeit very slowly. And the bigger the pieces of DNA, the more slowly they'd be able to wiggle through the tiny pores in the agarose molecules once the gel sets. It's just as if you had Jell-O and you mixed it with sugar and artificial coloring, right? The Jell-O effectively makes a sort of a matrix, a sort of a 3-dimensional net, and in between the links of the net, we have the molecules of food coloring and the molecules of sugar and we can mix those together to make our delicious Jell-O dessert. Here, instead of sugar and artificial flavors, we can have DNA, and we can move the DNA through the gel using electricity. That is because DNA has a negative charge. If we look at the sequences of DNA, this is a half of a strand of DNA, and on each side we see those phosphates, and the phosphates have a negative charge. So every rung of the DNA ladder, this is one strand, there's another strand on the opposite side. So every base pair is effectively -2, and if I have one base pair, I have a -2 charge, and if I have 2 base pairs, I have a -4 charge, and if I have 100 base pairs, I have a -200 charge. So the same mass to charge ratio in DNA makes any piece of DNA roughly the same in terms of mass to charge ratio. So because it's got a negative charge, if I apply a negative charge to this end and a positive charge to this end from an electrode, from a battery, or from plugging a gel box, a power supply into a wall, then DNA with a negative charge will avoid the negative electrode and be attracted to the positive electrode. It'll move through, yeah? And so as it moves through, big pieces will go slowly and small pieces fast. It works kind of like a jar of marbles. If I take a jar of marbles to the beach and I get some sand with big sand grains and little sand grains and big pebbles and little tiny pebbles and so on, and I dump the sand into the jar of marbles, the small grains of sand will reach the bottom first because they squeak through the spaces in between. The big grains of sand will take more jostling to get all the way to the bottom compared to the others, yeah? The space between the marbles would allow sand to fall, larger grains would fall slower, and so on. So in this case, the jar of marbles is like the agarose gel, and the DNA is like the sand, and gravity effectively is like the electric charge that would be ordinarily pulling the DNA through. Gravity is pulling the sand grains through. Okay, so we get our gel, we get little samples of DNA, we mix the DNA with a nice blue dye, we put the dye and DNA into the gel, we zap it, and then the DNA runs to the red, moves towards the positive electrode away from the negative electrode. And so what we see once that's done is little lines, and the little lines are pieces of DNA that are small down at the bottom, that are big up at the top. And those DNA fragments, the smaller they are, the faster they wiggle through the pores in the gel. Okay, so how do we use this principle to solve a crime or find out who the parent is? Well, or the male parent is, the daddy is, as was highlighted in that entertaining song that we just listened to. Well, we can take DNA samples and treat them with restriction enzymes, yeah? So at the crime scene, oh, we've got another little video here. Do you guys ever watch CSI? Me either, but I went and found some CSI. Those CSI people.
I rotate start to finish.
So we know that one woman is connected to both our Vicks, but without her prints, we can't connect her to Derek Cursor. Looks like we're stuck.
Maybe not. I'll pay a little visit to my horse buddy. See if he can identify Isabella as the woman in the shadows. If he
Boy,
can,
those people talk really fast.
It's like we're looking at the wrong thing. The artwork in Isabella's office.
wait a minute.
Isabella's self-portrait. It's the latest thing. We're looking at her DNA.
That's her DNA.
We made a
She's
pact.
busted.
None of us wanted to wither away, hooked up to tubes and machines.
Boy,
So we
what a
agreed to help each other die
fascinating
whether we
show.
wanted to die.
I don't know. It actually might be a good show. I've never actually watched it. But so there it is. We see DNA segments from an individual person forming a DNA, effectively a DNA fingerprint. So you amplify sequences that are different in people, yeah? People are 99% the same, but there are some regions of DNA where there's a lot of difference, yeah? we amplify those with PCR. We take the samples, we digest them with restriction enzymes, yeah? We take the DNA at the crime scene, yeah? Somebody left the perpetrator's blood, there was a struggle, and the perpetrator was cut. We sample, we find the DNA, and we amplify it. We treat it with restriction enzymes, the amplified sequences, yeah? And the DNA, we have an enzyme that cuts CCGG, and anytime it sees CCGG, it cuts. In this sequence, it cuts 2 times to make how many pieces of DNA? Well, if we have a linear piece, you cut it twice, you get 1, 2, 3 fragments. Here, it's ACGG, CCGG. So, does this cut? No. The restriction enzyme only cuts CCGG. So if it sees this, it doesn't cut it. If it sees this, it does. If it sees this, it does. And if it sees this, CCGG, it does. Yeah? So at the crime scene, DNA, we amplify it, and then it gets cut into 3 fragments. Fragment W, X, and Y. The suspects only gets cut into fragments Y and fragment Z, yeah? And when we run those out on a gel, fragment Y is in both, and that appears at the bottom. Why? Because it's the shortest segment. It's the smallest piece of DNA. It moves quickest through the gel. So the band, this band at the bottom is a bunch of little pieces of DNA, all of this size, yeah? This band is the same, a bunch of pieces of DNA of the same size, yeah? Fragment W is down here, and it's a bunch of pieces of DNA of this size, and this band here is the medium-sized piece. The large piece is equal to the sum of the small and medium pieces. This is the extra small piece. And so that is the Z line here, very big piece, yeah? And so, did the suspect do it? No, but we can get a high-priced lawyer to convict him anyway, right? Because, anyway, no wait, that's just in America. All right, so by sorting DNA by size, we can analyze and know how big the pieces of DNA are. So, can we answer this? Who done it? Suspect 1 or suspect 2? You should be able to tell. Okay, so that's restriction digestion. That's all I got to say about that. Pretty neat, don't you think? Okay, thanks for listening. Click here to